Hi, so my research is looking at the effects of settlement and habitat preference on eastern bluebird breeding biology. So a little bit about me. My name is Melissa Bailey. I am a senior biology major here at Truman. Once I graduate, I plan to go to grad school to study behavioral ecology. So this research is really in line with my interests. So a little bit of background information on why it's important to understand the factors driving settlement patterns. So the conditions that are present early in development can greatly impact quality and potential survival of an organism. So these conditions can be things like food or resource availability, presence of predators or competitors in a region, and temperature, and they can affect different morphological, behavioral, or physiological aspects of an organism, which can then impact its potential survival as well as its potential future reproductive success. So a few examples of this that we see in nature. The first is incubation temperature influencing metabolic rates of blue tits. So this was an experiment done in which the incubation temperatures of blue tits were either raised or lowered, creating three groups, and the resting metabolic rates of the birds within each of those three groups was measured, giving us this graph. So they found that lower incubation temperatures, as seen here, correlated to higher resting metabolic rates, whereas higher incubation temperatures, as seen here, correlated to lower resting metabolic rates. So this is an example of a developmental condition, in this case, nest microclimate, influencing offspring phenotype, in this case, in affecting the resting metabolic rates of the offspring. Another example is poor song production in adult song sparrows that experienced nutritional stress as nestlings. So this is an example that shows a developmental condition which causes a change that then is carried out throughout an organism's entire life. So this is an example that shows an effect which could potentially decrease an individual's quality over its entire life. In this case, the song production, which could limit its reproductive success, as song production in song sparrows is a factor driving mate choice. So both of these examples are showing the importance of developmental conditions as they can greatly impact a wide variety of traits in the offspring. So because developmental conditions are so important in influencing offspring traits, it's important to understand the factors that are driving parent choice, as there is a direct link between parent choice and the types of developmental conditions that their offspring will be experience. So the bird we were looking at in this research was the eastern bluebird. The eastern bluebird is a sexually dimorphic species, meaning that there is a notable difference in morphology between the males and females. So the males have a more vibrant blue coloration along their back and head, as seen in the top photo here, and the females have a more muted bluish gray coloration along their back and head, as seen in this lower photo. Their breeding season is February to mid-September, and they generally have more than one brood per breeding season. So in terms of their preferred habitat, they tend to prefer areas with limited woody vegetation that have a larger distance from perches or tree lines. They also tend to prefer open areas like fields that allow for higher levels of visibility from their box entrance. And while these are the optimal types of locations, they will nest in suboptimal locations if those are the only ones available to them. And then the reason that they're a good species to study when studying settlement patterns, they are secondary cavity nesters, which means that they are unable to create their own nesting cavities and instead must rely either on natural cavities or artificial cavities, so human-made nest boxes. They are also highly territorial, so there is competition both within the species, within other members of the species, as well as within other species such as wrens, house sparrows, and tree swallows. So both of these factors mean that there is a natural limitation on the number of high-quality nest boxes that an individual can occupy. So while the environmental factors of an area surrounding a nest can influence whether settlement will occur at that location, the aspects of the cavity itself can also impact whether settlement will occur. So these factors can be things like cavity orientation, so the location of that the box entrance is facing, whether it be towards an open field or towards a tree line. cavity size and depth, presence of old nesting material, and cavity age can all impact whether settlement will occur. So one possibility for a factor which could be impacting settlement and offspring survival is box age, so new versus old nest boxes. And there are several reasons for why box age would potentially be either positively or negatively impacted, impacting settlement. One possibility is that the presence of ectoparasites, so ticks or mites, could be negatively impacting settlement in older boxes, which are more prone to have higher levels of those ectoparasites. Another possibility is that pre-existing knowledge of older boxes in a location could positively impact settlement in those older boxes within a species. So here are a few examples of birds preferentially choosing new boxes over old boxes. This first was an experiment done with red cockaded woodpeckers. The woodpeckers were presented with the option of new versus old natural cavities and then new versus old artificial cavities, and they found that when given the option, they will nest in the newest possible cavity regardless of new or artificial. Another example is tree swallows preferentially choosing to nest in the newest nest possible when given the option. And the reason for this being that they are attempting to limit the presence of ectoparasites within their nest, as the presence of these ectoparasites could potentially decrease their offspring quality. So while those were examples that were showing preference towards new boxes, there are also examples of birds preferentially choosing older boxes. So this first example is an experiment done with eastern bluebirds, which showed that the eastern bluebirds preferentially choose boxes containing old nesting material when given the option between boxes with old nesting material and empty boxes. The reason for this being with a box that already contains nesting material within it, there is a decreased energetic cost expended in gathering new nesting material and building a new nest from scratch. Another example was a survey of secondary cavity nesters' nest reuse rates, which showed that within secondary cavity nesters, as shown here, reuse rates were 48%, which was far higher than reuse rates of either primary cavity excavators or weak cavity excavators, with birds of the same species reusing nests in subsequent years with very few skipped years in between. So birds of the same species were choosing to return to nests which provided success within successful breeding attempts from year to year. So while we know that there is a potential difference in settlement between new and old boxes, it's currently unclear how or if this impacts overall nestling quality. So the specific questions we are looking at are, does presence of new and old nest boxes impact settlement in a region? And how is nestling quality impacted by nesting in a new versus an old nest box? And then the specific comparisons we will be making between new and old nest boxes are how does settlement differ between new and old boxes, as well as survival, reproductive initiation, and nestling quality in terms of nestling body condition and nestling ornamentation. So boxes were made and placed from February to early March. We had 48 total boxes that we were monitoring across those 3 sites. So the Truman State Farm, which is shown here, had a mixture of old and new boxes. The golf course, which is shown here, had all new boxes, and the school, which is shown here, had all new boxes as well. Data collection and banding was done from March to August. The adults were captured and banded once their eggs had hatched, and measurements were taken of the tarsus length, which you can see in this photo, right wing length, which you can see here, mass, and then tail length, and photo and feather samples were taken as well. Nestlings were captured and banded once they were 14 days old, and measurements were taken of their tarsus length, right wing length, mass, and photo samples were taken from them as well. So in order to determine if there was a significant difference in settlement between new and old boxes, we ran a Pearson's chi-squared analysis, which tells us the probability of observed differences happening by chance. And in terms of settlement patterns between new and old boxes, we found that settlement was contingent on box age between all 3 sites, with birds preferentially choosing to nest at the farm, which was the site containing a mixture of old and new boxes, as compared to the sites which only contained new boxes. However, when looking within the farm, there was settlement was not contingent upon box age, meaning that while birds were preferentially choosing to nest at the site with both old and new boxes, they were not preferentially choosing one box type over the other. So we ran the same test, the Pearson's chi-squared analysis, to determine if if there was a difference in nest survival across the 3 sites, and found that across all 3 sites, survival was not contingent on box age. And then this graph on the right is showing the difference in reproductive initiation date between the new and old boxes, which is showing that on average, individuals which nested in old boxes began laying eggs far earlier in the breeding season as compared to those individuals nesting in the new boxes. So body condition or body quality of the parents was calculated by graphing a best fit line of the mass and tarsus measurements taken in the field. So the residual of the graph, so this distance here from each point to the line, was used as our metric for body condition. So a negative body condition, which is shown here, which falls below the best fit line, would indicate a lower quality individual, whereas a positive residual, which is shown here with the green line, would indicate a higher quality individual. And then the same process was used to determine nestling body condition at each nest, with each residual representing the average body condition for all nestlings in one unique nest. So this graph is showing difference in adult body condition between the old and new nests, which is showing that on average, there was a higher level of body condition for those individuals nesting in old nests as compared to those nesting in new nests. And then this graph is showing the same relationship, but in the nestlings, which is showing that body condition between the new and old nests was comparable in nestlings. So for color analysis of the birds of each nest box, this process is still currently ongoing. Currently, we have analyzed the photo samples of the nestlings and adults taken in the field using a program called ImageJ. So for each photo sample using ImageJ, we were able to generate histograms such as these shown on the right, which give us quantifiable data about different aspects of the coloration of each bird. And these aspects are things like hue, saturation, brightness, and intensity. So still to do in this research, our next steps are to complete the analysis of the physical feather samples taken from each of the adults, and to complete the analysis of the color variation between new and old boxes to determine if there is a detectable difference in coloration between the new and old boxes. I'd like to thank the Wilson Ornithological Society and the True Scholars Research Program for providing me funding to do this research. I'd like to thank my research mentor, Dr. Hubbard, as well as Dr. Goldman, who allowed me the use of his nest boxes. I'd also like to thank all the members of the lab who helped me out with this research. Thank you very much.